I'm just watching as people are readily coming in. I know we have a guest tonight who probably needs no introduction, but our speaker is a MacArthur Fellow and Distinguished McKnight Professor of Entomology at the University of Minnesota. Her research efforts focus on protecting the health of all bees, breeding hygienic bees for their natural defenses against disease and parasites, promoting sustainable beekeeping practices, and propagating flower-rich, pesticide-free landscapes to support nutrition, health, and diversity of bee pollinators. Please join me in welcoming Dr. Marla Spivak.
Thank you, Lisa.
Thank
Hi,
you for
everybody.
joining us.
How's it going for you? This is a interesting year and interesting time, and I wish everybody the best of health for the rest of the year, and whatever outcome of the election you want, I hope it goes well. So I'm going to talk to you. I'm going to share my screen. And I think that while I'm doing that, I can't see the chats, but I'm going to stop periodically and see if you have questions. Or Lisa, if you see things going on, go ahead and interrupt me.
Sure.
It's nicer if we can have more of a conversation in this 2D format that we're all in these days. So I'm going to share my screen, and you should be able to see it now, right? Okay, good. So I'm going to talk to you about something that I've worked on all my career. So I got a job at the University of Minnesota in 1992. I became interested in hygienic behavior in 1990, 1991, when I was a postdoc at the USDA lab in Tucson. And I was working with Martha Gillum and crazy Steve Tabor. Maybe some of you remember him, eccentric guy. But they both got me very interested in this behavior. And so I'm going to kind of review a lot of the older research and then bring you up to date. And what you're looking at on one side is the actual, can you see my pointer? Yeah. Bees doing the process of hygienic behavior, which is removing problematic brood from the nest. Here's a little piece of it over here. And then a diagram of it when it's in relation to mites. All right, that didn't work. Let's try. There's got to be a better way to do that. My thing wasn't working. We'll figure it out. So let me zoom out a little bit and give you some context for what hygienic behavior is. A woman whose photo I'll show you in a little bit from Austria named Sylvia Kremer coined this term social immunity in 2007. And in English, what it means is how social insects maintain the health of their colony. When you think about it, I mean, you're seeing a honeybee, really dense society, ants that live in really dense society, termites, here are leafcutter ants, but it would be true of social wasps too, the yellow jackets and hornets that live in really dense societies. And we should all relate to this right now. If a disease or a pathogen or a parasite moves into the colony, how do they keep themselves from how do they keep it from transmitting throughout the nest? Think COVID, think big cities, think nursing homes, think how do we prevent disease transmission when we live in close quarters like that? Her definition was the behaviors, organizational strategies, and physiological mechanisms that social insects use to defend against parasites to maintain the health and survival of the colony. So basically how they keep themselves healthy. And here she is, this is Sylvia Kremer. She works on ants. And she said, she called it there's two strategies of social immunity. She called it care kill, but the word kill is pretty strong. So I'm going to use the word eliminate. It's a little gentler. Humans, we tend to take these words and put human values on them, but when they're pertaining to social insects, it's a little bit different. But let me explain to you what she thinks. She studies ants in these little Tupperware containers. The whole thing is in the laboratory. So she keeps colonies of ants, but they're in the laboratory stacked in these incubators and these containers. And they're tiny little ants. And what she's observed them doing is they have in their care department, it's like preventative medicine or prevention, right? That's what care means to her, what the ants do. And that's just to prevent getting a disease. So they would groom each other. They might have antimicrobial secretions. I'll show you that in a minute, and just keeping the nest clean. But once they're infected, they move into the eliminate strategy. And then what they sacrifice individuals that are sick and infectious. So I'm going to stick with this ant example because it's really cool. And as beekeepers, maybe you can have some fun knowing a little bit about these ants. So here's the ant over here. It's Lasius neglectus. What a great name, huh? And she can paint mark some of them to watch them. They get a disease that's a fungus. It's called metarhizium. It's a soil-borne fungus that lives in the soil. And since ants live in the soil, they're really subject to getting fungal pathogens on them and in them. But what they do is if they get a little bit of this metarhizium on their pupae, they groom the fungal pathogen right off of the pupal silk cocoon. So they're constantly grooming off, keeping their pupae clean. If they also secrete a little bit of venom and put it right on the silk cocoon of the pupa, and that disinfects the fungus. So venom is antimicrobial. It's very understudied in honeybees, I should say. We should be looking at the value of venom. Bees do spread it on their bodies, honeybees do. And are they using it as an antimicrobial and as a disinfectant also? If an ant, a pupal ant, does get infected, so the care strategy didn't work, but they get infected and the fungus moves in and starts killing the pupa, what they do is I need to explain. The fungal spores are what attach to the pupa, and they're infectious. Spores are highly infectious. Think AFV spores, for example. They're infectious. But then the spores germinate, and it's a fungus, they start growing these mycelia. The mycelia are not infectious while they're growing inside. But after a while, the mycelia then produce spores again. So for a while the fungus is invading the pupa, it is not infectious, it's not contagious. And so, but the pupa gives off this cue, probably an odor to the adult ants. And it says, hey, I'm sick. And so the ants take off the cocoon completely, the silk cocoon. They tear apart the ant pupa and they inject all the parts of the pupa with venom, again, to disinfect it. And then they throw it into a chamber in their nest where they keep dead and trash. They can't throw it outside the nest like a honeybee would do because ants, or maybe you don't know ants. Ants would go outside the nest, oh, here's something, and bring it right back inside the nest. Even if it's been disinfected, they don't want it to be brought back in. So they put it in a special chamber where no one except a few ants just go in, deposit things. So it's pretty cool. So those are examples of the care eliminate strategy. So for honeybees, what are they? Well, I would argue that preventative care would be propolis collection, which I could talk to you forever about, but I won't today, unless you ask. And propolis is antimicrobials. Bees, as far as we know, don't produce any secretions that are antimicrobial, but they collect resins from trees that are antimicrobial. And their elimination strategy is hygienic behavior. So once infected, they start using hygienic behavior to remove infected individuals from the nest. Okay, so what is hygienic behavior? I don't know where that arrow came. That's pretty fun. The definition, and it was discovered early on in the United States with Park and Woodrow in the 30s and 40s, and then later by Rothenbuehler in the 60s. And so we've known about this behavior for a very long time, but it's the ability of bees to detect and then remove or eliminate the diseased brood, and again, before the disease forms the infectious spores. So just like with the ants, the bees are detecting there's a problem with the pupa while it's non-infectious, okay, before it goes back to the spore stage. And so it's a behavioral defense against American foulbrood and chalk brood diseases, 2 diseases of the brood. Questions, real quick, before I move on?
So, quick question, and how do they, do we know how they detect the disease?
Yeah, you have to stay tuned though.
Okay, all right, fair enough.
All right, I'll just keep going. The other thing that I've stressed now, and I want to keep stressing, is that hygienic behavior in it, it has critical detection and timing components. And this was discovered early on in the 40s with Woodrow and Holst, these researchers that were in, I think they were in Wyoming at that time. There was a USDA bee lab in Wyoming back then. They published that resistance to American foulbrood in the honeybee colony consists in its ability to detect and remove the diseased brood before the causative organism reached the infectious spore stage. So you can imagine, here's American foulbrood, and this right now, when it's brown and goopy and stinky, or when it forms a scale, it's highly, highly infectious. So the bees need to get this thing out of the cell before it reaches this stage in the right photograph. So they're going around, and I'll explain how they detect it. Well, I can tell you, it's by odors. They detect it very early, before we can even see that there's symptoms. The bees are very sensitive at figuring it out and remove it. Huh, it's really funny how I got a little red arrow in everything, right? These little gremlins move in and take over my slides. So another point and I'll bring this up at the very end again. We started selecting bees for hygienic behavior, and we bred this line we called the Minnesota Hygienic line. And I'm going to walk through all these steps here. The first thing you do is assay a bunch of colonies. You present them with freeze-killed brood, dead brood, and I'll go through that process. And then once you find colonies that you deem to be really hygienic, you have to propagate them. And we used instrumental insemination. This is an insemination device. And then it's always very, very important after you've had these inseminated queens that you actually test them with a pathogen or Varroa mites or a parasite to see if they in fact are resistant. So let me explain this. Early on in the 40s, when they were looking for hygienic colonies, they would actually do that challenge first. They would actually give a colony disease and then observe what happened. But given a bunch of colonies, American foulbrood is kind of tough, because then you got to burn everything at the end of your experiment. So researchers came up with this idea of just testing the colony by presenting them with dead brood. And then in my lab, we kind of refined the very sophisticated technique. You can see we use PVC tubes here, 3-inch PVC. We just twist gently right into the comb. And then we take 400 mils, which is about 10 ounces of liquid nitrogen, which you can get at a welding store. You can rent a Dewar tank, and don't put it in a regular thermos, you'll blow it up. But we use liquid nitrogen and we pour it right down that PVC. And then once this is thawed, you can gently remove the PVC and put it back in the colony. So there's about 160 pupae within that circular PVC pipe. So the deal is that when you see how quickly the colony removes dead brood, it's correlated kind of with how quickly that colony might remove diseased or mite-infested brood. So it's a way to really screen a bunch of colonies just testing them with dead brood so that you don't have to give them the actual pathogen first. So what you're looking for after 24 hours is a colony that uncaps and removes all of that freeze-killed, that dead brood within 24 hours. So this one we would call really hygienic, really, really rapidly hygienic. And that's what you're really looking for. Most colonies, and I would say about 85% of the colonies everywhere, are more like this, where within 24 hours, they're getting at it. You can see they're starting to uncap, but they haven't really removed all of it. And this is about 50%, and some of them are a lot worse. So what we do is go around and test colonies on a really large scale, right? So this is our liquid nitrogen tank. It's fitted with a valve here. You really, really should wear gloves. I don't know why I still have these photos when you're using liquid nitrogen is like minus 300 something. It's what they use to remove warts. So maybe this guy just was trying to do some wart removal at the same time. Anyway, so we pull frames. This would be out of 1, 2, 3, 4, 6 colonies, put a PVC in there. We're recording the number of empty cells that are empty or have larvae in them to begin with, or the number that are completely sealed. And then you can see, we usually mark with a tack or an X on the top bar. You can see after we remove the PVC, we put it back in the colony, come back in 24 hours and count how many are left. Okay, and then we subtract from 160 and you can do the percent removed. And it's really important to repeat it twice. So once we find colonies that remove, we're looking for those that remove 95% or more within 24 hours. And once we find them, we raised queens from those colonies and I inseminated them with semen from drones from other hygienic colonies, right? So that's how we bred a line. And I kept track of all the pedigrees so that we would avoid inbreeding and, you know, hundreds of colonies and pedigree lines. It's very complicated, but here's one of our queens. They're kind of derived from Italian stock. You can do this from any stock. All stocks of bees show a certain percentage that are already hygienic, and it's just a matter of finding them and propagating them. Okay, so I've taken you through the assay and then the propagation. And now I'm going to show you some of the step 3, the challenges, when we actually then give the bees that were bred a challenge and see what happens. So early on, what we did was we found a colony that had really bad American foulbrood, and we kept the frames in our freezer in the lab. And then we cut up frames, sections like this, so that they were just full of AFB scale. And we went into perfectly good hygienic colonies and some that were not hygienic. And we cut out a section of their middle brood comb and we put in the scale right smack in the middle of the colony and then let the colony rear brood. And then over time, we'd quantify how much AFB, how much American foulbrood they got, because this scale is really infectious. So this was one circle of the apiary, and can you guess which ones are the hygienic colonies and which ones are the non-hygienic colonies? These are not, they were not making honey, and there's another one between there you can't see very well. But. So some of the data, so the hygienic colonies are in blue, and these are the weeks that we observed them and the number of colonies that came down with American foulbrood. And so you can see by week 2, there were 6 out of the 18 colonies total of the hygienic colonies that got some American foulbrood. But over time, over 3, 4, or 5, by the end of the experiment, there was 2 colonies that had AFB. And in contrast, almost all but 1 actually of the non-hygienic colonies got American foulbrood, and it kept up. So the ones, these 6 that came down to 2, they got over it through hygienic behavior, through the constant removal of the diseased brood. But the non-hygienic bees leave it in there and it just becomes more and more infectious. And if I'm not showing you here the severity score. So those that did have, the hygienics that did have American foulbrood had a really, really light infection. and these had a really bad infection. And then the end of this story is we had to burn it all. And I want to emphasize that these non-hygienic bees are interesting because all bees uncap and try to remove sick and dead and dying brood. All of colonies do this. It's just how fat or how slow some go and how fast others go. So if you're really slow at it, so here's a colony with chalk brood, and you can see they're opening up the cell, but this is a chalk brood mummy in here already with spores and infectious. And you can see this American foulbrood. You can see the bees have started to uncap here. But if they try to remove this, it's super, they're just going to spread the disease around. So either you get it before it's infectious and very quickly, or if I were a bee, I would leave the cap on and don't touch it ever again. And just kind of entomb it in there. But they tend to open it up, which is not such a good strategy. So in about 1994, we started asking, is hygienic behavior a defense, one of the defenses that bees might have against Varroa? So we had this hygienic line that we were breeding, and then we would collect Varroa from colonies, and we would slightly uncap a cell, just lift up the cell capping and take a Varroa mite on a paintbrush and slip it in the cell and then tamp the cell capping right back down, and then mark where we put it in on a plastic transparency. And then we would come back over time to see how many of the cells the bees had figured out that we put a mite in there, and they opened it up and then would remove the brood. You with me on that one? When you introduce mites into a cell like this, you have to do it within 12 hours of the cell being capped over with wax. So what we do really is take this transparency and go to a colony where there's larvae, you can see them, that are just being capped, and we mark those and then come back in a few hours. And those are the ones that we introduce the mites into. Otherwise, if it's too late, the mite won't reproduce correctly. So you have to put the mite in as close in timing as they would go in naturally. Sorry. So again, the hygienic colonies here are going to be in this bright blue, and the non-hygienics are going to be in turquoise. Control means that we just simply opened up the cell capping and put it right back without introducing a mite. And the solid ones here are when we would open up a cell, stick in a mite. And this is at the end of 10 days after introduction, what percent of the mites, what percent of the cells or the pupae were removed by the bees. And so in 1994, the hygienic bees removed almost 70% of the pupae that we put a mite into. And this is about just over 60 and about 60%. This year, I have no idea what happened. We couldn't figure it out, so we started putting 2 mites per cell in, and when we did that, this number went way up again. But this year is just an odd year. But the non-hygienic bees, you can see all the years, this one's a little higher, they didn't remove very many of the mite-infested brood. And the controls, you know, sometimes the hygienic bees are opening up cells that we didn't even put a mite into. So they can, very sensitive to the disturbance.
Marla, is the hygienic behavior a specific role, or is it shared by all of the workers, commonly referred to as nurse bees?
Right. The bees that do the behavior are they're middle-aged bees. So they run from about 12 days to 20 days old. They're not the youngest bees, which is kind of nice if you think about it, because if you're going to set out a group to remove sick brood, you don't want them to be the nurse bees that are actually then feeding the brood. It'd be nice to have a little dividing so you're not accidentally infecting some of the younger larvae. But the bees that do the hygienic behavior do all the other tasks too. It's not like that's the only thing they do in life. You know, they'll go secrete wax, or they'll, whatever the middle-aged bees do, receive nectar from the foragers, and they're doing all the tasks. That they're just also kind of sniffing around looking for brood that has a problem. We could tell by the timing of removal that the hygienic bees were mostly detecting and removing the mite-infested brood after the mite started laying eggs, initiated oviposition. So you're looking at a pupa that's getting pretty well cooked. It's like cookies cooking in the oven. They're starting to turn dark here. Here's the eye, here's the mite, here's the mite babies, here's the male, and these are mite offspring. This one just went through its last molt and it's starting to get the adult coloration of the mother, okay? But as soon as earlier in the development of the pupa, when it's still white, as soon as there's a few offspring in the cell, the bees really detect that there's something wrong and they start opening up the cell and they remove the whole pupa, okay? They dismember it, just like the ants. They dismember it. Sometimes they eat it, or they throw it out of the nest. And sometimes this mother mite, the foundress, can escape during the process. So she may go around and jump back on an adult bee. There she is. You can barely see her here in the diagram. But the bees kill all the offspring. So for the mother mite, it's a zero-sum game. She's gone in there, she's laid her load of eggs on that pupa, but then the pupa's gone, the babies get killed, the baby mites get killed, and it takes her a little while before she's ready to lay eggs again. So her reproductive cycle is interrupted, and overall there's less mite reproduction in the nest. Got that?
Is the hygienic behavior a learned behavior? Is it a genetic trait?
It's a genetic trait. They do seem, no one's really looked at this, but they do seem to kind of get primed, if you will. So if you keep giving them dead brood day after day, they get quicker and quicker at removing it. But overall, there's colonies that do it very quickly and colonies that are very, very slow, and everything in between. So what you're doing is just trying to find the colonies that are really quick at it, at detecting and removing. We did a lot of field trials with commercial beekeepers. We would give them hygienic queens. They'd put them in their colonies, hundreds of them. And we would look at honey production, and we'd look at disease and mite loads, just to try to convince beekeepers, especially the large scale commercial ones, that there's no problem. If you select for hygienic behavior, not selecting for some other trait like aggressive behavior or something that you don't want. So they all produce as much honey, if not more, than unselected stocks. And they all had significantly lower disease and mite loads compared to unselected stocks in the commercial beekeepers' apiaries. All right, moving forward. So that was our research up until about 2007, 2009, basically. And then the Baton Rouge lab came up with this other line of bees that eventually they called Varroa Sensitive Hygiene, or the VSH. Should say VSH, not VHS. Okay, the VSH line. But first, I'm going to go back a little bit and explain how that line came about, because it's confusing to a lot of people. So John Harbaugh was a researcher at the USDA lab in Baton Rouge, a great researcher. He's retired now. But what he was trying to do was find colonies that didn't have very quick mite population growth over the season. So along with Roger Huppengarner, who was at Michigan, they got a bunch of colonies and they measured their mite loads early in the spring, and then they just left them alone. And then they measured their mite loads again in like August. And they were looking for colonies that didn't have very many mites by August. So they kind of ran a mite race, if you will, and the colonies that grew the most mites, they threw out, and they kept the colonies that didn't have very many mites by August. And they kept breeding from those. and throwing out colonies that would have a lot of mites by the end of the season and keeping the colonies with few mites. And they raised and inseminated queens from them. And they didn't understand how those colonies were keeping the mite levels low. So they called the line suppression of mite reproduction, because they thought somehow the bees were preventing the mites from reproducing. So they called it the SMR line originally. And then I asked John Harbaugh if I could have some of his queens, of his SMR queens. This was in the middle, about 2004, 2005. We introduced them into colonies. And, you know, at that point, I, you know, like if you're, what's that saying? If you have one tool, you always use that tool. I don't remember what the saying is, but so if you have a hammer, you use the hammer for everything. Well, our tool was this freeze-killed brood assay. So once I had these SMR colonies, I tested them with the freeze-killed brood assay and found they were like really super hygienic, more than my hygienic line that we were breeding. And then we put mites into cells just like we did before. And that's this graph that I'm showing you. So these are mites into SMR colonies, mites into hygienic colonies, and their controls. And you can see after 10 days, the SMR line removed, uncapped and removed much more of the mite-infested brood than our hygienic line. So I told John Harbaugh, hey, I think the reason they're not reproducing is because they're hygienic. They're removing all the pupae with mites in them. And so he looked at that also and did some experiments and published it with John Harris in 2005. And based on that, they said, okay, this line that we thought the bees were actually suppressing the mite reproduction was actually, they're hygienic. And so they named the line Varroa Sensitive Hygiene. Now I have it right, VSH. Okay. And so this line is now available. They have a new line now, the POL line, POL, for pollination, and beekeepers are liking that one better than the VSH. VSH sometimes is a little quirky in its behavior, but the POL line is a very nice line. But they're also hygienic. But the way they select for it is not through the freeze-killed brood assay at all. They don't use that down in Baton Rouge. What they do is very tedious, and we've done this here, and I'm glad I have the freeze-killed brood assay. So they take a colony they want to test, and they take a frame into the lab under a microscope, and they uncap 100 or 200 cells in these rows like this, and they determine what the % mite infestation in the brood is. So if you do uncap 100 cells and you see, oh, there's 10 mites in there, you're 10% infested, right, in the brood. And then they would take that same colony and put it in their VSA or their test colony and leave it in there for 10 days, and then remove it and do the same thing again. Go through on that same frame another 100 or 200 cells and compare the % infestation. And if they're the VSH, this hygienic VSH, they would have less infestation afterwards than they went in, because they were removing the mite-infested brood. You can also see there's uncapping. They tend to uncap a lot of the brood. Okay, and that's how they've done various methods, but this is the most common way that the Baton Rouge lab has used to develop the VSH. And for this reason, it's very difficult for beekeepers to do this. It's not difficult to do, it's just so tedious. You drive yourself nuts under the microscope going through all those combs. There's a lot of confusion among researchers whether the hygienic, and this is to show you that there's confusion in my head. That's what that's all about. There's a lot of confusion about whether the hygienic line based on the freeze-killed brood test and the I did it again. I look at this, I'm dyslexic with that line. The VSH line, the Varroa sensitive line, are they the same trait or are they different traits? Some people in the Baton Rouge lab, they say it's Varroa sensitive because they think the bees are only queuing in to mite-infested brood. And that the Minnesota Hygienic line is what they call general hygienic, and because it will detect disease brood and some mite-infested brood. But the VSH line detects mite-infested brood, and they're much, much better at it. Okay, so they think they're really specific. The behavior is specific to mites. But what hasn't been done is what I'm going to call the acid test. And that is that we should challenge some of these VSH colonies with a pathogen. Let's give them a little chalk brood, or let's give them some American foulbrood, and do they uncap and remove that also? If they do, then they're the same traits, right? If they don't, then it is Varroa specific and sensitive hygiene. And why we haven't done that particular test, I don't know, mostly because it's awful to give colonies American foulbrood, but should do it. All right, here's the answer to your question. And we spent about 10 years trying to figure this out. How do these bees detect disease in mite-infested brood? And so we ran 2 or 3 PhD students in different kinds of tests. We paint marked every bee in the colony to figure out what age the bees are that do the hygienic behavior. We would collect them, the right age bees that were performing the behavior, and we'd run them through an electroantennogram where you stick an electrode in the tip of their antenna. This is the ground electrode. You blow odor across them, and then you can run it through a GC and a mass spec to figure out what odors they're actually detecting with their antennae. Their antennae are their nose, right? So what odors can they detect? And then we'd present those odors to the bee in these learning trials. It's called proboscis extension learning. I won't go into the whole thing, and ask the bees if they could distinguish between the odor of dead brood and the odor of live brood. And the hygienic bees were always super good at determining distinguishing the difference between dead or chalk brood infected brood and healthy brood. And the non-hygienic bees were awful at it. We looked in their brains. This is a stain of part of the bee brain, looking at neurotransmitters that might be involved in odor perception. And so we just ran them through the gamut. And what we found is that the bees are detecting odors with their antennae, and that hygienic bees are able to detect and discriminate between the odors of disease and healthy brood at a really low stimulus level. So non-hygienic bees detect the dead brood, but only when it's like screaming stinky. And if you think about this for a minute, if you walk by your refrigerator in the kitchen, if you're a hygienic bee, you're going to smell that cheese that's rotting, or that's in your fridge, even though the door is closed and it's in the cheese compartment, right? That would be a hygienic bee. You're so sensitive to that odor. A non-hygienic bee would be one that the cheese is in there, but you have to open up the refrigerator, and you got to open up the cheese, and you're still rifling around, and you still can't figure out where that huge odor is coming from. You have to get right in front of it in order to really smell it. Does that make sense?
A couple questions. Does the odor they're detecting from the disease, is it from the disease, or is it from the state of the brood?
That's a good question. We don't know. So we thought it was actually the brood, but we used chalk brood to determine the odors. But we could never figure that out. There have been a lot of research more recently with the mites, and they think, the newest research that's not even published yet, they think the odor is coming from the immature mites, not the mother mite even, not the pupa. They think it's coming from the baby mites, which kind of explains why the bees start detecting after the mother mites start laying eggs. But they even in that research can't tell if it's really coming from the brood that gets into the baby mites or the other way around. So don't know yet.
When the cell is uncapped and the pupae is removed, can the mother mite lay more eggs at that time?
Yeah, she's refractory for we don't know how long, a few days maybe, but then after a while she can go and she'll lay another batch of eggs. She can do that 4 or 5 times in her lifetime, I think. So more recently, so we haven't been doing work on hygienic behavior in my lab since about 2009. We moved to propolis and the effects of propolis, the benefits of propolis to bees. But other researchers, this one's from 2015 and 2016, they're looking at the same thing, but they're all looking at mite-infested, Varroa-infested brood. And again, they're at least confirming that we found that the antennae hold the key to VSH behavior, that and there's specific cues associated with mite-infested brood. Okay, Alison McAfee, is that her name? Allie McAfee from Canada. Her PhD work, she found that when it's mite-infested, Varroa-infested, the bees can detect the problem with a brood ester pheromone coming from the pupa. And then once the cell is opened, what triggers removal is this other compound they're calling P32, which is this really long chain hydrocarbon. So this is the one that would be volatile, saying something's wrong. And then once they uncap it, they say, oh God, something is really wrong. So 2 different odors. If it's freeze-killed brood, she thinks that the first odor they're getting from dead brood is beta-ocimene. And once they uncap it, they're getting oleic acid, which is notorious compound. It's the smell of death for bees and ants and other insects. They produce nerolic or oleic acids when they're dying. Kara Wagoner, who is in Olav Ruppel's lab in North Carolina, she did some really tricky experiments. She took hygienic colonies from the hygienic line, VSH, and a control, and then she had queens lay eggs on all these frames and then chopped them up and reassembled them, put them back together, bound them together. So this one would have brood from hygienic, VSH, and control, hygienic, VSH, and control, each one. And she put this one into hygienic colonies, this into VSH, and this into control. So she gave each of these kind of colonies brood from all the other colonies, right? The other kinds. And she found that the hygienic behavior is influenced by a specific chemical coming, and she thinks it's from the brood, not the mites, and that it differs depending on the breeding program. So she thought the Q, the chemical odorant coming from hygienic line, was different from the one coming from VSH line and control line. So I have to tell you that this research is ongoing. It's really conflicting. We don't have the answers yet. It's confusing. I'm really glad I backed out when I did, because it was clear to me at the time. But now it's gotten more confusing and it's still in progress. But I wonder, people, this is again, good, this time now my little arrow pointed to my confused head. That's perfect. It worked out one time. I wonder, instead of looking for one particular chemical or one odor, one compound, I wonder if bees just know the odor of healthy brood. So think about it. If you're a bee, you're constantly walking over these brood frames and your antennae are touching the larvae, they're touching the capping over the pupae. And most of the time it must, to them, just feel, it smells good, 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 good. And then, ooh, something is not right. It's abnormal. And this is actually how our immune systems work. They can detect something foreign, not by a particular compound, but by the lack of self, if you will. And how nestmate recognition works. In other words, how bees determine who's in their colony and who's a robber or who's a foreign bee. And ants do this too. So they can just detect something different. So I'm just throwing this out there as a research idea for all of you who want to do a research project, that I wonder if instead of a particular compound, it's just the lack of healthy odor that they're detecting. I don't know. All right, so I'm going to stop, but the take-home points are, for those of you who want to do this freeze-kill brood test, and you don't have to do it with liquid nitrogen. I can explain other ways to do it. And if you're looking for these colonies that are super hygienic, it's really important that somebody, a researcher, actually challenge them, give them mites or give them a disease to see if they really, really are hygienic, because you get a lot of false positives and you get some false negatives with the freeze-killed brood assay. It's not a perfect test. So I'll take questions. Maybe I'll stop sharing my screen, although I can come back and share it later if I need to.
One more general question. Do you know, has any progress been made in getting the bee factories that sell packages to use more hygienic queens?
Yeah, okay, yes, good question. They are doing the hygienic tests, at least the package producers in Northern California. I don't know if the ones in Georgia or the Southeast are. The ones in California, many, if not most of them, are doing the hygienic test. The problem is when you have a hygienic queen, that queen needs to mate with a bunch of hygienic drones. So our calculations is that if a queen mates with 10 males, half of them need to be from hygienic colonies in order for her colony to be hygienic. And I think what's happening is the queen producers are good at raising these hygienic queens, but they're mating with who knows who are not getting the trait.
Marla, a number of years ago, we used to have a QRI, Queen Rearing Initiative. You know, husbands and wives have a difficult time staying together. Try to get 4 beekeepers to get along all the time. They put in a lot of time at the apiary. In any case, we had bought a number of VSH queens over a few year period. They weren't inexpensive. So I hesitate to think that any package providers are going to put $175 queens inside the packages.
No. Those that you're buying are intended, they're called breeder queens. You're supposed to raise daughters from them.
Yes, that's what we were doing.
Yeah, they're not going to sell them in packages as production queens.
No.
My hope would be that we could encourage the queen producers to just pay attention to the drone colonies. So if they're raising hygienic queens, why don't they take their own daughters, those good hygienics, and requeen their own stock? Because those queens will produce good drones, right?
Yes.
Coming straight from the queen's eggs. So if you have a good hygienic queen, the drones they produce are also going to carry the hygienic trait. So if we could just encourage them to requeen their own stocks with more hygienic stock, they could saturate the area with really good hygienic drones too.
Well, that's what we were aiming at, like starting from a central location and getting everybody that had bees within a certain range.
Yeah.
To take some of these queen daughters that we were selling. It's difficult though. You do need quite an operation. A lot of nukes going on there and a lot of dedication. So it sort of dwindled away over time.
I get it.
Another thing that we were trying to promote was everybody treat simultaneously for Varroa. If they were going to treat with formic acid, Mite-away quick strips was the medication du jour at the time. So we were trying to get people to sort of coordinate through the club to all treat at the same time so that we didn't have, what do they call them, mite bomb apiaries that just, whether you were treating or not, if your neighbor a quarter mile away wasn't treating, they were going to come back for you.
There's a question on the chat. What's the overwintering success of these hives without treatment compared to non-hygienic hives or with or without mite treatment?
So we never let them go without treatment, but I can tell you that we only needed to treat once in the fall, and not all the colonies really needed to be treated. Their mite levels were pretty low. When we put them in the commercial apiaries, then the mite levels would get much higher there because we were comparing them in the same yards with some non-hygienic stock. And we found that when they got to about 10–15% infestation, the colonies would still collapse. So they never survived without treatment, but we never really gave them that test either. We are starting another breeding program here at the University of Minnesota, and it's a little bit different. Do you want me to talk about that just briefly? Do you have a second, or am I over time, or how are you doing there?
No, go right ahead.
I have this new idea. So a lot of people are promoting the survival stock, the James Bond method, live and let die. So you just get a bunch of colonies and don't treat them. And then next spring, you might have a colony or two that happen to survive, and you try to breed from those. And I'm never really like that because it's a big genetic bottleneck. You're losing a lot of your genetic diversity, but also you have no idea why those colonies are living. And, as a scientist, I kind of want to know. And so we're trying this, we're going to try it. I'm at I have 5 more years before I retire, and I have nothing to lose. And so I thought I'm going for it. So what we're doing is we have a bunch of colonies. We call them the parent colonies, and we make splits from them, lots of splits from each, like 3–4 nukes from each one in the spring. The nukes, so we're keeping really good track of the pedigrees, the lines, the mother colony, the parent colony, and the nukes that we make from them. The nukes we don't treat. We're wintering them as nukes. We wrap them together. They're what we call 5/5. So a 5-frame nuke with a 5-frame super on it, if you will. And we wrap 5 or 6 nukes together for the winter. We just did that today. And we don't treat those. But I keep the mother colonies, and we're treating those as they need it. And we're doing lots of different measurements on the mother colonies: hygienic behavior, propolis collection, mite population growth, mite reproductive success, grooming behavior. We're doing a million measurements on the parent colonies. And so that way, when the nukes, those nukes that do survive this by spring and they're alive, I can figure out, I'm hoping to figure out why they're alive from the data from their mothers, the parent colonies. Does that make sense?
Yeah.
So in other words, what I'm trying to do is follow the bees' lead, because I think they know what they're doing better than I know what I'm doing or what they're doing. To develop resistance to mites, instead of trying to drive them with one and select for one particular character, I think there may be multiple things they're doing to keep their mite levels low. So I'm going to let the bees lead me, but through the nukes, but through their parent colonies, I hope to figure out where they're leading me. Does that make sense? Stay tuned. We're in like year 3 of this.
No other questions?
I'll throw one out there, Marla. First of all, thank you for taking the time with us. This has been great. And I'm actually really excited to hear about the outcome of these new studies. Question about, so how long or how many generations are required for this trait to be adapted within the species?
Let's see, maybe what you're asking, because I may not be understanding your question, so I'm going to rephrase it. How many generations did it take to really get the hygienic trait into the line? Is that what you're asking?
Yes.
It takes 4 or 5 years. So the first generation, when you have a good hygienic queen and you inseminate her with good hygienic drones, you look at her progeny, her daughter colonies, and some are hygienic and some are not. You got to keep selecting, and it takes a good 4 years before you really start to see it level out and the colonies get pretty even homogeneous in their level of hygienic behavior.
Interesting. That's pretty quick though, when you think about evolution, right? And how traits evolve in a variety of other species.
Yeah, but it's quickly lost if you let queens open mate with any old drones. It goes away like that.
Those damn any old drones. How long is it going to take? For example, the parasite does not necessarily want to kill its host. So how long is it going to take for the bees to develop a symbiosis with these Varroa? Like,
They would do it quickly if if there weren't any beekeepers. Sorry about that, but
no,
it's true, because we keep bees in dense apiaries,
yeah,
or close together, or in cities where there's lots of bee colonies, and then there's drift and robbing, as you pointed out, and so in that way, that evolutionary idea that I don't need to kill my host, or I shouldn't kill my host, goes out the window, because the mites can always find another colony to go into.
yeah. Now the Africanized bees, apparently they've developed a behavior where when the mite load gets too high, they're going to abscond. They're going to just go find a new home, leave all the old mites behind, begin to the increase of the mite load at their new home, and then comes a point where they take off again. Inhospitable hive, they're gone. That's how the Africanized, my understanding is that's how the Africanized bees have developed.
Well, they're in part, they have much smaller colonies.
Yep.
They are on the move a lot. They swarm a lot, so they're dividing themselves a lot. They
Yeah,
also
they're running away from the mite.
and they also abscond if their nest gets too infected or infested. Yeah, so they have lots of different ways to keep mites.
Even our colonies will abscond over a few day period if the mite load gets too high. We've been noticing that with the chit chat, but amongst the group. I went out.
Partly that's viruses too. So viruses direct behaviors of organisms, of us, of bees. And what the virus wants is to get into more bees, right?
Yeah.
And so when a bee has a lot of virus, the way they die is fly out of the nest, and they probably try to get those bees to fly into other nests so they can continue to infect.
A couple questions in the chat. Let's say you do not test for hygienic behavior, but evaluate your colonies year round and pick the best 10 to 15% of queens to use them as breeder queens. Are selected hygienic queens any better than the queens raised by selecting the best 10 to 15%?
Well, it depends on what you call the best. So the ones you really like, they still may be loaded with mites. But I would also say that it brings up a good point that I should have brought up during the talk, in that we would select using the freeze-killed brood assay. But if we had a daughter colony that we didn't like, had an attitude, testy, nervous, we got rid of them. So we were always culling colonies that we didn't like to work with. So we were ending up with really nice, gentle bees, and they had to winter well in our area, and they had to fulfill a bunch of other criteria for us, not just be hygienic. But if by best you mean those with the lowest mite levels, that would, yes, you can definitely make progress that way. But in order to really, this is a good group project for you. You need a bunch of microscopes, I think, or you need young kids with really good eyes. And that isn't me, but you take a sample of mites on adult bees in May or whenever, and then you go through 100 cells of brood and you say, what % have mites in them? Okay, and that's your start. And then you do it again in August. You collect your 300 bee sample, and you shake the mites off in alcohol or powdered sugar. So you get your mites on 100 bees, and you open up another 100 brood cells. And you try to get the stage of pupae where they have a pink or a purple eye, so kind of middle stage of development, kind of all the same age. You go through and you see the percent mites in the
That's
brood. And when you do that, you compare it spring and fall, you get so much information, because sometimes you see colonies, let's say they have 5 mites per 100 bees in August, like mid-August on adult bees. And you look in the brood and they have fewer percent mites in the brood than they do on adults. That would tell you they're hygienic. Other colonies have 5 mites per 100 adult bees. And you look in the brood and it's like 70% of the cells you open up have mites in them, and sometimes 2-3 mites per cells, and babies galore. And you just go, talk about a mite bomb. As soon as that brood emerges, man, boof, that colony is going to be dead. And you can really get a lot of information, but you got to dig in the brood, which is
Marla, I use a dental pick, and I have gone to people's places and I've picked out the brood. I have seen 2 and 3 mites per larva. Is that considered 1 cell with a mite, or is it considered 3 mites?
Well, there's 3 foundresses in there. They all climbed in. Those are really juicy larvae, and you toss that colony. Those colonies should be cold.
Okay.
Because the bees should if there's 2-3 mites in there, the hygienic colonies would remove that. Odor would be so strong.
No, I popped one a couple of years ago, and I just pulled about 20, and there were quite a few larvae, and they had multiple mites on them, and the mites were cordovan colored, so they were ready to start coming out.
Yeah, those are the ones you don't want.
Can you clarify why you're not treating the nukes again?
Because I want to see which one of them survives. But from the ones that do survive, I can trace them back to their mothers. I know the mother colony I made the nuke from and all her characteristics. So I might be able to figure out why those nukes survive. So it's survival stock, but I'm also running the parents in parallel to tell me why those nukes are living, what traits that genetics has. That's my idea. Sorry.
Thank you. Is there an environmental or geographic angle to the hygienic trait? Like, why were the bees from the south better than your bees, or is it just simply
It's
genetics?
simply genetics, but it can speed up or slow down. So when bees are in a nectar flow, a big honey flow, all of them are more hygienic than when there's not a honey flow. So when you're testing colonies, you have to test them at the same time. And then if you retest them, you compare at that time. So you can't compare like June when there's a big flow, or July. You can't compare the results to August when there's a dearth or something.
Another question, when mating, queens will look for a DCA up to 9 miles away, whereas drones seek one around 5 miles as a mechanism against inbreeding. Has any work been done to place apiaries over a wide area, taking this mechanism into account?
It's usually not quite so far. Usually the drones are going, the queens are flying farther than drones from their own apiary, but they like to go half mile or a mile. And the drones like to go like a 1/4 mile to a half mile. And sometimes the DCAs are like right above your apiary. I don't know if you've ever seen them, but they can be pretty close, not necessarily. And so yes, the commercial queen producers have areas where they have their mating nukes, and then 1/4 mile, half mile in a big area around that, they have their drone mother colonies. Most of them do that in California anyway.
Have the genes responsible for hygienic behavior been identified?
There's many genes, and I'm going to say no. many attempts and many claims that they have the number of genes, but every time they do it using a different technique, it comes up with a new number. What they do know is the genes that they found are associated, most of them with olfaction, smelling, but not always.
Do you do single drone insemination or multi-drone insemination method?
Well, it depends. Usually it's multiple drones. I put in about 8 to 10 microliters, which is the equivalent of about 10 to 12 drones worth of semen in every queen. A single drone insemination would be for a specific experiment. The new breeding program in Hawaii, they're doing a lot of single drone inseminations. That gives you the trait right there. So if you have a queen and one drone with traits you like, the progeny from that you can breed from, because that's all you have. I mean, there's not a lot of variation. The problem with these single drone inseminated queens is they don't live and lay very long.
Did you find any areas in the hive where hygiene was better than in others?
No, if they're really hygienic, they're good at it everywhere.
Lisa, did Fatih have a question that didn't get responded to?
I don't think so.
I thought I saw some writing.
Questions to asked.
Okay. All right, I'm gonna go get a beer. Anybody want one?
Any final questions? We'd like to thank you so much for taking the time and meeting with us today. We really appreciate it. It's just a wonder of Zoom that we said, let's reach out and make it happen. So we really appreciate your willingness.
Happy to do it. I put on my Zoom sweater for you.
Thank you, Marla.
You're welcome. It's great to hear you. Great to see you all.
Thank you
Good
so
luck.
much.
Thank you.
Thank you.
Thank you.
You're welcome.
Have a great evening.
All right. Bye-bye. Thank you, Lisa.
Okay,
you.
folks, you don't have to go home, but you can't stay here. Bye.
Bye,
All right,
Chuck.
I'm gonna leave. Bye-bye.
Thank you.
How about some apple
Lisa.
pie?
Yes,
Lisa, thanks for organizing that. That was great.
thank you. And I said, let's see if she'll come, and happy to have her here. It's wonderful.